A very good morning to one and all. Myself, Ananya Bishnoi, and here I am very grateful for receiving Best Researcher Award under the category of International Popular Scientist Award. So, thank you for selecting me as a Best Researcher Award, and I want to thank to organizing committee of Science Father and the Science Father too for selecting me for this award, and for my I also want to thank my supervisor, Dr. Gunjan Vasant Bonde, for his continuous guidance throughout this journey. And here I'm going to present on the topic Electrospun nanofiber, the versatile platform as a drug delivery system in healthcare. So here I begin with the contents of my slides. First of all, I will brief about the overview of nanofibers, then electrospin setup, featuring features about the nanofiber, material for electrospinning, and the application of nanofiber in healthcare, such as diabetic foot ulcer, cancer, microbiological disorder, wound healing, cardiovascular diseases, and tissue engineering. And lastly, I'll end up with the conclusion. So here I introduce that pharmaceutical research with novel drug formally include formidable challenges, and only 11% reaches at the market level. So there are many challenges for the researchers, and these anti-cancer drugs have improved potency. However, their intrinsic toxicity continues to raise concern. So here we come up with the nanotechnologies like microsphere, sphere nanoparticles, myceli, nanogels, and nanofibers. So, which is a helpful and innovative approach in drug delivery approaches. Biodegradable and biocompatible polymer-based nanofibers have gained much attention because of their effectiveness, unique physiochemical attributes like extensive surface area and small diameters. So, nanofiber scaffold mitigates the downside of systemic drug delivery, thereby enhancing pharmaceutical actions. Nanofiber that we have uh, shown in this paper that it can be fabricated by drawing interfacial polymerization, force spinning, melt blowing, phase separation, template, melt extrusion, template synthesis, and electrospinning. So here we are only focusing on the electrospinning technique because the electrospinning technique is advantageous due to its relative ease of use and adaptability and versatility for producing a range of biomimicking nanofibers for a wide variety of polymeric and inorganic materials. So there are some other advantages of electrospinning techniques such as during the nanofiber preparation, such as pharmaceutical research involved novel drug include formidable challenges and merely 11% reaches at the market level. So low cost and simple equipment is necessary. Scalability for industrial production is required. Control over nanofiber diameter, porosity, composition, and alignment is required, and it has the ability to incorporate drugs or signaling molecules into the nanofiber. It has the ability to modify nanofiber mesh by post-electrospinning processes, serve as a drug delivery device, offering controlled and sustained release of drug with high drug loading efficiency. By adjusting the electrospinning parameter, the release profile and degradation rate of drug-loaded nanofiber can be modified. So there, I am expressing about the electrospinning setup. In the electrospinning process, there is a liquid drop which dynamically changes and form a jet that is then stretched around. There are four parameters for primary components for electrospinning, like spin rate, which is a glass syringe, high voltage power supply, electrically conductive collector, which is a piece of aluminum foil or silicon wafer to collect the fabricated nanofiber. There is a syringe pump which provides the constant flow of spinning solution. So the procedure include that the electrically conductive electrospinning solution is loaded into a 5 cc glass syringe. DC voltage is applied. Electrospinning solution acquires a stable shape between electric charge and surface tension. Later on, this increasing voltage form a Taylor cone, which when electric charge overcomes surface area. Finally, the equilibrium breaks between surface tension and electric charge. Ultra fine nanofibers are formed. So here we can see in the picture as well that there is a pump, a power supply, syringe, Taylor cone, and also the polymeric solution, which is forming a ultra-fine nanofibers. So what are the features of electrospin nanofiber? Electrospinning has become widely employed technique for nanofiber as tissue engineering due to many reasons, like they offer a high porous structure and large surface area that facilitate rapid cell signaling and attracts fibroblasts for the secretion of extracellular matrix component. They mimic functional structural characteristic feature of native extracellular matrix, supporting cell attachment and proliferation. The interconnection between the pores in electrospin nanofiber helps protect wounds from microbial infiltration. Also, it provides the controlled and sustained drug delivery devices with a high drug loading capacity, further enhancing the potential of a wound dressing. So, what are the material for electrospinning? They are basically various material, but I am here defining few materials like organic polymer, small polymers, composite materials. So what are organic polymers? Organic polymers are the polymer that dissolve in suitable solvent to produce solution or can be melted without degradation. Most of these polymers can be directly used in electrospinning. Then we come up with the small molecules like when chain enlargement is strong enough to stabilize the electrifying jet and so reduce the railing instability, small molecule can directly electrospin into fibers. Example here it is lecithin, which is the first natural mixture amphiphilic small molecule. Then we come up with colloidal particles. A dispersed phase along with the dispersion medium makes up a colloid system. These systems are now optimized for electrospinning when enough particle enlargement has formed to maintain the jet's continuous structure. So the composites are the Respun, which are usually produced by mixing nanoscales or solution gel precursor with polymeric solution. The gelation, condensation, and hydrolysis of the precursor should be started in the jet during electrospinning by coming into contact with the surrounding air. This causes the inorganic phase to form a continuous network in the polymeric matrix, which results in nanofiber spinning of an inorganic polymer composite. So here are the applications of in healthcare. Like we can see nanofibers, nanofiber generated using electrospin are flexible platform that may be managed drug release rate with sustained and controlled drug release, employ polymers with inherent healing capacity, load biomolecules and nanoparticle efficiency, modulate physiochemical and mechanochemical behavior, and more advantages are also there. So we are going to discuss about few of the applications in healthcare like wound healing, cancer, biotherapy, biosensor, cosmetics, artificial organ, tissue engineering, cardiovascular, foot ulcer, or pollution removal. So the very first example is diabetic foot ulcer. Diabetic foot ulcer is characterized by nerve damage, that is neuroaxonal degradation or demethylation, leading to the impairment in nerve conduction. So there's a study which came up with the deferoxamine DFO-loaded nanofiber for wound dressing and which promote the angiogenesis and healing of the wounds. So the researchers fabricated DFO-loaded biofilm nanofiber mats from biopolymer like chitosan and sodium alginate and other polymers like polyvinyl alcohol by electrospinning. DFO promoted the wound healing by regulating hypoxia indicible factor 1 and improved the stability. So the prepared nanofiber met the desired standard of the supreme wound dressing nanofiber, which have the high degree of swelling and appropriate water evaporation transmission rate, greater entrapment efficiency, and sustained drug release with inhibition potential against gram-positive and gram-negative bacteria. Also, the bilayer nanofiber mat was discovered to be non-cytotoxic, evident as in vitro scratch test demonstrated the wound healing capacity, which indicates that the material is effective for the DFU, diabetic foot ulcer. So the polymeric nanofiber mats can prove to be better delivery system for wound therapy in the diabetic foot ulcer. So here is the another application in tissue engineering, where we can see the same image of SEM image of tissue engineered polymer that were formed. So tissue engineering is a technique that includes isolating healthy cell from patient and then exploiting them in vitro processes. So there's a researcher Andres Hurtado that he reported that a line fiber graft enhanced vascular CNS tissue regeneration. The author grafted astrocytes loaded unidirectional aligned and random fiber into rats, thoracic spiral cord, and separated by 3 millimeter of gap. So conclusively, I am stating here that aligned PLA fiber had proven significantly fast recovery of nerve tissue injury as compared to random nanofiber by fast migration of astrocytes and regeneration of axons. Another application is wound healing. So wound healing is really important because chronic injuries are challenging to heal on their own, owing to damage ECM and loss of residual stem cells, which results in researchers to want to create a 3D biodegradable scaffold which mimics natural ECM to substitute defective or missing ECM. So there is a study which collected liquid collecting electrospinning was used to successfully build polylactic acid co-glycolic acid PLGA based 3D fibrous scaffold with 520 micrometer sized interconnection pores. The goal for the using of natural ECM component to modify the surface is to provide biological recognition for cell proliferation. After 7 days of culture, human dermal fibroblast effectively scaffolds to a bottom of 1400 micrometer and demonstrated considerable gradual proliferation on collagen type immobilized scaffold. Such findings suggest that 3D fibrous scaffold might be used for chronic wound healing. Another one is filtering rate for microbes and particulate matter using E. coli bacteria as a prototype microorganism and electrospun polyurethane chitosan nanofiber as a filter medium. The researchers studied the impact of various elements on the diameter and homogeneity of these nanofibers. After preparation, he suggested that the ideal circumstances, effectiveness of chitosan and polyurethane electrospin nanofiber filter in filtering out nanoparticle was assessed. So with the increased polyurethane polymer solution concentration, mean diameter of nanofiber increased while their homogeneity decreased. And therefore, according to the bacterial disk diffusion result, electrospin produced polyurethane chitosan nanofiber exhibits significant antibacterial properties against the model bacterium E. coli. So the spinning produced chitosan urethane nanofiber exhibits significant antibacterial properties against the model bacterium E. coli. Another here we deal with the cardiac complications, which were reported in the previous studies, like the heart pumps blood through the body. Impaired cardiac function arise from myocardial ischemia, which is brought by the metabolic accumulation and hypoxia inside the heart rate. So designing these cells is now possible because of biomaterial synthesis, nanofabrication, and use of appropriate scaffold having different topographies and architecture. So researchers from different researchers discussed a variety of redeemable polymers, including natural polymers, synthetic polymer, combinatorial polymers that may help in creating fibers. They also talked about several fiber modification approaches that by mimicking in microenvironment element like the ECM can enhance cell migration, proliferation, survival, etc. The function and variation of nanofiber-based heart regeneration scaffold were also discussed, and additionally uses of scaffold and stem cell infusion research method were improved with improved biocompatibility and restoring the cardiac muscles capabilities. Next, I'm covering the cancer therapy. So there are quite drawbacks to administer anti-cancer medications, both orally and intravenously, including limited effectiveness, poor solubility, and so on. So a dual delivery system was created in one demonstration which combined RNA interference with chemotherapy for the treatment of brain tumor. Such example contain matrix metalloproteinases 2. Expression of glioma signal pathway was precisely suppressed by complexing a Taylor RNAi plasmid complex with polyethylene amine gene carrier. So RNA interference plasmid and paclitaxel, which is a cytotoxic drug, were then encapsulated in PLGA fiber, inducing a long-term release for both the drugs. Invasive and angiogenesis of brain tumor were decreased by the suppression of matrix metalloproteinase 2 expression, while the development and protein proliferation of cancer cells was stopped by cytotoxic drug paclitaxel. So the study showed that the use of synergistic treatment resulted in a considerable increase in efficacy when compared with the individual drug. Another, I'm going to deal with the cosmeceutical part of healthcare. The development of cosmeceutical product with natural bioactive substances had captured the attention of cosmetic business, which has led to the recent development in the use of nanofiber in cosmeceutical industry. So there is a study which combined pomegranate and tea tree oil extract, which were encapsulated using the electrodynamic technique and added to facial beauty lotions. The formulations were kept in storage at three different temperatures, including those with addition. Then the using the dynamic mechanical analysis approach, rheological experiment, and so on, they decided and assessed that changes in storage modulus, complex viscosity, and loss modulus. So dynamic mechanical study revealed that all formulation could be used in cosmetic business, and that modification brought on by storage time and temperature had minor impact. So next, I'm dealing with the biosensors. Electrospun nanofiber have employed extensively in the field of sensors with a high sensitivity and quick reaction time because of extremely porous structure and the high surface to volume ratio. The nanofiber can detect change in concentration of chemical species, including tiny molecules like water, glucose molecules, and even gaseous species. For instance, like porous TiO2 nanofiber with 30% of lithium chloride topping demonstration high sensitivity to variation in humidity. The sensitive identification of compounds that are crucial in maintaining human physiology has also received a lot of attention. Another, I am dealing with the microbial disease. So there are nanofibers application in healthcare system critically deals with microbial diseases. It encounters the limitation which are already available treatments like vulvovaginal candidiasis can be treated with Marvico, acyclovir, and azithromycin in a blend of PLA, providing 100% release in 1 hour. Due to messy consistency and grainy resistance of the vaginal drug delivery system, urges the design of appropriate formulation. Mucoadhesive formulation require addition of plasticizer for quick along with greater antifungal properties. So here I am concluding my presentation that nanofiber has achieved impressive success in areas of drug delivery system and drug administration and transitioning. Conclusively, it is important to note that prolonged effectiveness of using prolonged drug delivery system is increasing in healthcare sector with the advancement in wound healing, tissue engineering, artificial organ, and so on. Because of the enormous potential, scientists from many fields are working together to address the obstacle, and there are future perspective that there might be more chances to overcome such limitation which have with the already available treatment. As a result, the drug delivery industry, which has expanded quickly during the previous 10 years, does not appear to be facing any major obstacle. The idea of bringing the gap is ongoing, and commercialization may happen shortly. So here are the bibliographies and the researches that I have covered. Thank you.